maintenance of the balance within the gut microbial composition and fermentative process is thus essential for health. For example, we know for a long time that elimination of hydrogen, the main gas produced from fermentation, this elimination is essential to maintain efficiency of the fermentation. This is assumed in the ecosystem by a specific group of microorganisms which are called hydrogen-consuming microorganisms. Any disturbance or alteration of this elimination of this hydrogen removal could induce important metabolic shift and alter the fermentation process and products. More recently, several works have showed that there is this disruption of this intestinal microbiota in several pathologies such as obesity, type 2 diabetes, or inflammatory bowel disease. The microbial mechanisms involved are not well known or are still under study, but it seems that this microbial mechanism varied according to the pathology considered. For example, in obesity, the disruption in the gut microbiota leads to an increase in the energy harvests available for the host. This is probably due to alteration in the fermentative process. Microbial disruption could also be involved in the onset and maintenance of another pathology which is called irritable bowel syndrome. Irritable bowel syndrome is a gastrointestinal functional disorder characterized by abdominal pain or discomfort associated with modification in bowel habit. Alteration in the gut microbiota composition and stability has been already reported. We recently demonstrated that the gut microbiota of IBS patient is characterized by an important functional dysbiosis affecting different metabolism, which are metabolism of butyrate, metabolism of hydrogen, and also metabolism of lactate. And this dysbiosis is really characterized by the predominance of a specific group of microorganisms which are called sulfate-reducing bacteria, that are bacteria which produce sulfide. So the overall change in this gut microbiota of IBS may lead to important change in metabolic output and important change in fermentative process, and in particular to an increase in sulfide production. Sulfide are known now as a gasotransmitter that can facilitate somatic or colonic pain. Sulfides are thus bacterial metabolites that are a good candidate to be involved in visceral hypersensitivities. Thus, the gut microbiota of IBS is a good example of the role of alteration of gut metabolism in the pathophysiology.